My name is Hélène Gilbert. I work at INRA in France, and I'm here to present the results of the FIDOGENE project at the EAAP in Tallinn. The aim of the work package 5 in FIDOGENE is to put together new traits and new statistical models for selecting feed efficiency and bring them to the breeding programs and the breeders to improve the selection of feed efficiency. Our work package is very integrative as compared to the work packages in FIDOGENE in the sense that it brings together new measurements for feed efficiency that are produced in work package 2, for example, with new conditions for feeding the animals that are explored in other work packages and statistical models that we work on in work package 5. One of the main targets for selecting for feed efficiency is to work on traits that will be measurable on farm and that will be heritable. The idea of this is to have criteria that have a variability that is transmitted from one generation to the next and that is relatively cheap to measure so that we can record it on a maximum number of animals and select very accurately for the trait we want to select. Another target that we recently developed for selecting for feed efficiency is the selection or the study of gut microbiota as a potential new heritable trait. Until recently, we did not have any tools to measure gut microbiota individually in the animals, but with the advance of genomics, we do have new tools, so we are able to characterize the individual gut microbiota for selection. Our last target is to develop advanced statistical models. What we call a genetic model is a model that will be able to separate out the variability of a trait which depends on the genetics, so it is transmitted from one generation to the next, and the variability of the trait that is not transmitted. And the genetic models that we propose, for example, target the fact that animals are produced in groups, so they have their own performance, but the performance is also affected by the effect of their pen mates. In terms of outputs, they are relatively classic in terms of types of outputs that we produce for animal breeders. There are new measurements on new criteria to better select for feed efficiency, components of feed efficiency or components of robustness to avoid to impair it while selecting for feed efficiency, and new methods that the breeders will be able to implement in their selective program later on.